students, my name is Niyati Said and thanks for watching Wikipedia word videos. My topic for the presentation is cancer. Cancer is a class of disease characterized by out of control cell growth. As you can see, the unlimited growth of cells is known as cancer. Okay, there are over 100 different types of cancers and each is classified by the type of cell that is initially affected. Okay, cancer harms the body when altered cells divide uncontrollably to form lumps or masses of tissue called tumors. Okay, except in the case of leukemia, where cancer prohibits normal blood function by abnormal cell division in the bloodstream. Tumors can grow and interfere with the digestive, nervous and circulatory system, and they can release hormones that alter body functions. Tumors that stay in one spot and demonstrate limited growth are generally considered to be benign. More dangerous or malignant tumors form when two things occur. First, a cancerous cell manages to move throughout the body using blood or lymphatic system, and thus it destroys healthy tissue in a process called invasion. Second, that cell manages to divide and grow, making new blood vessels to feed itself in a process called angiogenesis that we will be discussing in detail. Okay, so let's proceed towards our topic. Cancer are a result of uncontrolled cell division, as I have told you, and that these can occur in any organ or tissue. In cancerous cells, control of mitosis has been lost and they divide, and this process goes on and on and on. Okay, this leads to tumor, as I have told you, that displaces healthy tissue and interferes with proper function. Cancer can arise from any cell capable of mitosis. Okay, what is tumor? Tumor is a cancer tissue, you can say. Okay, now the question arises, how do cancer cells differ from normal cells? In normal cells, hundreds of genes intricately control the process of cell division. Normal growth requires a balance between activity of those genes that promote cell proliferation and those that suppresses it. Okay, means there is a balance between cell proliferation and cell suppression. Okay, which is all controlled by genes, of course. It also relies on the activities of the genes that signal when damaged cells undergo apoptosis. Now, what is apoptosis? Apoptosis is a programmed cell death. Okay, cancer cells have the cells that gone wrong. In other words, they no longer respond to many of the signals that control cellular growth and death. You may say that divides indefinitely and they no longer respond to many of the cell signals that control cellular growth and death, of course. Okay, and second, cancer cells originate within tissue and as they grow and divide, they diverge ever further from normalcy. Okay, means they deviate from the normalcy. Okay, over time, these cells become increasingly resistant to the control that maintains normal tissue. Okay, that means they become very resistant to the cellular signals that control cellular growth and death. Okay, or you can say or that maintains normal tissue, and as a result they divide more rapidly and more quickly than their progenitors. Progenitors means initiators. Okay, and become less dependent on signals from other cells, as we know that cell is controlled by the cell cascade. Okay, that means cancer cells, they become less dependent on the signals from other cells. Cancer cells even evade programmed cell death. Programmed cell death is apoptosis. Okay, despite the fact that their multiple abnormalities would normally make them prime targets for apoptosis. In the late stages of cancer, cells break through normal tissue boundaries and metastasize. Metastasize means spreading of the cancer cell to the new sites in the body which are not invaded yet by the cancer cells. Okay. This is the last stage of cancer. We'll be studying stages of cancer in detail, and this is the last stage of cancer where the cell breaks through the normal tissue boundaries and metastasize to the new sites in the body. Okay. Now, we very know that cancer is controlled by genes. In fact, any normal cell is controlled by genes. Okay. Cancer gene. The cell division process is dependent on a tightly controlled sequence of events. We know that cell division process is dependent on cell cascade. Okay. These events are dependent on the proper level of transcription and translation of certain genes. As we know that process of transcription is when formation of RNA takes place from double duplex DNA, then that is known as transcription. And what is translation? Translation means protein synthesis. Okay. When this process does not occur properly, that means when transcription and translation they don't occur properly, then unregulated cell growth may be the end result. Okay. And of the 30,000 or so genes that are currently thought to exist in the human genome, there is a small subset that seems to be particularly important in the prevention, development and progression of cancer. Okay. These genes have been found to be either malfunctioning or non-functioning in many different kinds of cancer. The genes that have been identified to date have been categorized into two broad categories depending on their normal functions in the cell. Genes whose protein product stimulate or enhance the division and viability of cell. Viabilities means survival of the cell. This first category also includes genes that contribute to tumor growth by inhibiting cell death. That means it suppresses the cell death and thus it promotes the tumor growth. Tumor is a cancer cells. Okay. Genes whose protein products can directly or indirectly prevent cell division or lead to cell growth. Okay. The normal versions of genes in the first group are called as proto-oncogenes. Oncogenes means cancer genes and proto means prior to that. Okay. The normal version of genes in the first group are called as proto-oncogenes. The mutated or otherwise damaged versions of these genes are called as oncogenes. Okay. Proto-oncogenes, they are present in every individual, but when it's get mutated or otherwise it's damaged of version of proto-oncogene, then that proto-oncogene gets converted into oncogenes. Okay. Note that by convention gene names are italicized and proteins they make are not. Okay. As an example, P53 refers to the gene and P53 refers to the protein. Okay. The genes in the second group are called as tumor suppressors. Okay. The one which suppresses the cancer cells or the tumor. Now the question arises, how do cancerous changes arises? Gene mutations accumulate over time as a result of independent events. Okay. Consequently, the path to cancer involves multiple steps. Okay. In fact, many scientists view the progression of cancer as a microevolutionary process. You can say that cancer staging is a process of determining the extent to which cancer has developed by spreading. Contemporary practice is to assign a number from 1 to 4 to a cancer, with 1 being an isolated cancer and 4th being a cancer which has spread to the limit of what the assessment measures. The stage generally takes into account the size of a tumor, whether it has invaded adjacent organs, how many regional or you can say nearby lymph node it has spread to, if any, and whether it has appeared in more distant locations or not. That means metastasized. Okay. Overall, stage grouping is also referred to as Roman numeral staging. That is, this stage uses numeral 1, 2, 3 and 4. Okay, to describe the progression of cancer. There is also a stage known as stage 0. Okay, as you can see, this is a normal growth cell. That means it is a stage 0 or carcinoma in situ, and this then it progresses into the stage 1, in which the cancers are localized to one part of the body. Stage 1 cancer can be surgically removed if small enough. Okay, this is known as hyperplasia. Okay, and then comes a stage 2. This is atypical hyperplasia. Cancers are locally advanced. Stage 2 cancer can be treated by chemoradiation or surgery. Okay, and then comes a stage 3, that is carcinoma in situ. Cancers are also locally advanced. Whether a cancer is designated as stage 2 or stage 3 can depend on the specific type of cancer. For example, in Hodgkin's disease, stage 2 indicates affected lymph nodes on only one side of the diaphragm, as you can see, and whereas stage 3 indicates affected lymph nodes above and below the diaphragm. Okay, the specific criteria for stage 2 and stage 3 therefore differ according to diagnosis. Stage 3 can be treated by chemo and radiation or surgery. Okay, and then comes a stage 4. This is known as microinvasive. Okay, the cancers have often metastasized or spread to other organs or throughout the body. Stage 4 cancer can be treated by chemo, radiation or surgery. Okay, within the T&M system, a cancer may also be designated as recurrent, meaning that it has appeared again after being in remission or after all visible tumor has been eliminated. Recurrence can either be local, meaning that it appears in the same location as the original, or distant, meaning that it appears in a different part of the body. Okay. Stage migration describes changes in the distribution of stage in particular cancer population induced by either a change in the staging system itself or else a change in technology which allows more sensitive detection of tumor spread and therefore more sensitivity in detecting spread of diseases. Okay. Now, we will study about the carcinogenic agents. Carcinogenic agents means the one that causes cancer, okay, or the one that causes indefinite growth of the cell. First is geographical influences. There have also been associations made between different geographical regions and particular cancers. As we know that Japanese men, they have stomach cancer 5 to 6 times higher, okay, than others, attributed to the consumption of fermented food. Okay, because Japanese men, they consume fermented foods more than others. Okay, likewise, breast cancer is 20 times higher among American women. Okay, because of the fat American diet, and the liver cancer is 10 times higher in Africa, which correlates with high rate of hepatitis B infection. Okay, Likewise, liver cancer may also be caused by aflatoxin. Aflatoxin we'll be studying in detail. A food contaminant produced by fungi. Okay, this compound is prevalent in grain stores in tropical and subtropical regions because moist grain is very good place for the fungi to live or to sustain. Okay, so these were the geographical influences, or you may say that those were the carcinogenic agents that causes cancer in particular population. Okay, and second is lifestyle. The impact of many environmental factors can be reduced by making healthy lifestyle choices. One of the most potent carcinogens in human is benzopyrene, a compound which is found in cigarette smoke. Okay, in fact, the tar in cigarette smoke includes both initiators and promoters. Means it initiates cancers or the indefinite growth of the cell, okay, which makes it especially more dangerous. Alcohol is a promoter of carcinogenesis. Carcinogenesis? Carcino means the one that causes cancer and genesis means formation. Okay, alcohol is a promoter of carcinogenesis in humans, as is asbestos. Okay, as it is seen that the workers that work in asbestos factories, they are very prone to cancer, okay, or indefinite growth of cell. Additionally, ultraviolet radiation from exposure to the sun or tanning beds is a powerful initiator in humans and is a cause of skin cancer. Okay, and the third cause or the third carcinogenic agent is drugs. We'll be studying about the aflatoxin here. Okay, tamoxifen is a chemotherapeutic agent that is used to combat or to control estrogen receptor positive. Okay, estrogen is a hormone in women. Okay, so it is used to control estrogen receptor positive breast cancer, and thus it increases the risk of endometrial cancer by increasing the rate of endometrial cell proliferation. Okay, for this very reason, long-term tamoxifen treatment is losing popularity in favor of aromatase inhibitors. Estrogen themselves may also be important in the promotion of tumor, particularly in most postmenopausal women. The one has crossed the menstrual stage. Okay, that stage is known as postmenopausal women. Receiving exogenous estrogen due to their ability to increase mammary and endometrial cell division rates. This is an area of active research also. There are factors that can predispose an unborn fetus to developing cancer later in life. These include exposure to radiation or the synthetic estrogen diethyl stilbestrol, okay, that is DES. Now, how the cancer progresses? There are 3 stages: initiation, promotion, and the progression. A compound that reacts with DNA, that is deoxyribonucleic acid, and somehow changes the genetic makeup of the cell is called a mutation. Okay, the mutations that predispose cells to develop tumors are called initiators, and the non-reactive compounds that stimulate tumor development are called as promoters. Okay, approximately 70% of known mutagens are also carcinogens. That means they are also cancer-causing compounds. A compound that acts as both initiator and promoter is referred to as complete carcinogen, because tumor development can occur without the application of another compound. First is initiation. We'll be studying in detail. Initiation is the first step in 2-stage model of cancer development. Initiators, if not already reactive with DNA, are altered, okay, via drug metabolizing enzymes in the body and are then able to cause changes in DNA. Since many initiators must be metabolized before becoming active, initiators are often specific to particular tissue types or species. The effect of the initiators are irreversible. Once a particular cell has been affected by an initiator, it is susceptible to promotion until its death. Okay? Means after initiation it have to cross promotion, and then it have to cross progression also. Since initiation is the result of permanent genetic change, any daughter cells produced from the division of the mutated cell will also carry the mutation. In studies of mouse skin carcinogenesis, a linear relationship has been observed between the dose of initiator and the quantity of tumors that can be produced. Thus, any exposure to the initiator, it increases risk, and this risk increases indefinitely with the higher level of the exposure. And then this is the illustration how normal cells gets converted into the neoplastic cells, okay, or the cancer cells. This is the additional genetic alteration. This is initiation, this is promotion, and this is progression. When the initiator harms the DNA of the normal cell, then it gets converted into initiated cell, okay, and then it gets converted into pre-neoplastic cell, and the phenomena is known as promotion, and then ultimately it gets converted into a neoplastic cell, and this phenomena is known as progression. Okay, and the second stage is promotion. Once a cell has been mutated by an initiator, it is susceptible to the effects of the promoter. These compounds promote the proliferation of the cell, giving rise to a number of daughter cells containing the mutation created by the initiator. Promoters have no effect when the organism in question has not been previously treated with an initiator. Unlike initiators, promoters do not covalently bind to DNA or macromolecules within the cell. Many bind to receptors on the cell surface in order to affect intracellular pathway that lead to increased cell proliferation. There are two general categories of promoter. The one is a specific promoter that interact with the receptor or in the target cell of the defined tissue, and the second is a non-specific promoter that can alter gene expression without the presence of a known receptor. Okay, promoters are often specific for a particular tissue or a species due to their interaction with receptors and are present in different amount in different tissue types. While the risk of tumor growth with promoter application is dose dependent, there is both a threshold and the maximum effect of promoters. Very low doses of promoter will not lead to tumor development, and extremely high doses will not produce more risk than moderate level of exposure. This is the illustration how the cell gets promoted. These are the UVA, B chemical and the viruses that initiates the normal cell, and then it becomes an initiated cell, and then initiated cells get converted into definite growth, and thus it upgrades to a stage known as promotion stage, which has a higher cell proliferation, altered gene expression, and epigenetic changes are also seen, and then it ultimately upgrades to the stage known as progression, where the chromosomal abnormalities is seen, oncogenes activation is seen, tumor suppressor gene activation is also seen, and thus it causes skin cancer. Okay, now the third is progression. In mice, repeated promoter application on initiator exposed skin produces benign papillomas. Most of these papillomas regress after treatment is stopped, but some progress to the cancer. Okay, the frequency of progression suggests that papillomas that progresses to cancer have acquired an additional spontaneous mutation. Okay, The term progression, coined by Leslie Fowles, referred to the stepwise transformation of a benign tumor to a neoplasm and to malignancy later stages. Okay, progression is associated with the karyotypic changes, since virtually all tumors that advances are aneuploid. That means they have a wrong number of chromosomes. This karyotypic change is coupled with an increased growth rate, invasiveness, metastasis, and alteration in biochemistry and morphology. There are 4 stages, as I have already told you: stage 0, stage 1, stage 2, 3 and 4. Okay, that I have already discussed it with you. So now come to metastasis. Metastasis is the last stage, literally meaning new place, is one of the terminal stages of the cancer. In this stage, cancerous cells enters the bloodstream or the lymphatic system and travels to the new location or the new sites of the body, where they begin to divide indefinitely and lay the foundation for secondary tumors. Okay, when man reaches to a metastasis stage, then it becomes very difficult for him to survive. Okay, not all cancer cells are metastasized. In order to spread in this way, a cell must have the ability to penetrate the normal barriers of the body so that they can both enter and exit the blood or the lymph vessel. Okay, steps of metastasis. There are a few steps of metastasis that include: first, the separation from the primary tumor, and second, when it invades through tissue around the initial lesion and penetration of their basement membranes, and third is the entry into the blood vessels and survival within the blood via blood vessels, called as hematogenous spread. Okay, entry into lymphatics or peritoneal cavity, it is spread via lymph channel known as lymphatic spread, and it reaches the distant organs like lungs, liver, brain, bone, etc. Okay, and then it forms new lesion. Okay, along with the new blood vessels that feed the tumor, because tumor also needs feeding for the survival. Okay, and this formation of the new blood vessel is termed as angiogenesis. Okay, now come to the routes of metastasis. There are 4 possible routes of spread of cancer. These include spread via lymphatic channels. This is favored by most carcinoma. Spread via blood vessels. This is favored by sarcomas and some carcinomas that originate in the kidney. Because of their thinner walls, veins are more frequently invaded than arteries, and the spread is via veins. Okay, they also spread via body cavities. The cancer seed onto peritoneal cavity. Okay, and the pleural and the pericardial cavity, or the subarachnoid spaces, or the space that covers the brain. Okay, and third, by the transplantation of the cancer. This occurs by the carriage of fragments of tumor cells via needle or surgical instruments to other parts of the body during surgery and diagnostic procedures. Okay, this is the step of the process of metastasis. This is the normal epithelium, and where the cell grows as a benign tumor in the epithelium, and then cell becomes invasive and enters the capillaries or the veins. Okay, and then it adheres to the blood vessel wall in the liver. Okay, and then they escape from the blood vessel to form a micrometastasis, and then they begin to colonize liver, forming full-blown metastasis. So this comes to an end. I hope you liked my presentation. Thank you and stay tuned.